Let's move on to the main part of the talk and discuss the bioimage analysis workflow, starting with the basic rules to follow when acquiring images. We will start with the basic rules to follow when acquiring images. When you're acquiring images, you should ensure they are of suitable quality, because without good data, you won't get good results. Or you can spend too long trying to optimize your analysis for bad data to compensate for these poor quality images. There are 5 main rules you want to follow when you are acquiring your raw microscopy data. First, you should acquire with a sufficient sampling rate, avoid saturation, use a proper bit depth, use the whole dynamic range in the image, and use appropriate resolution. And I'll go through each of those in more detail in the following slides. To understand the basic requirements for image acquisition, we first need to establish what we mean by a microscopy image. So let's say these 3 spots represent the fluorescent signal from an object. These signals will pass through the optics of the microscope and form an analog image. When the signal is collected by the microscope camera or detector, it must undergo an analog to digital conversion so the signal can be processed by the computer. So what we see on screen will be a digital image made up of pixels, which are the basic unit of a digital image, and they will have a defined size. When we convert a signal from analog to digital, we can have different sampling rates. If it is too low, it may be too pixelated and we lose the details from our object. So first of all, make sure you're using the correct sampling rate by checking your acquisition settings to ensure your pixel size is suitable for the object that you want to image.
When the sampling rate is too low, this is known as undersampling. This will usually be apparent if you have a highly pixelated image that doesn't look like your sample. However, undersampling is even more relevant if you plan on quantifying your images. First thing you should do is determine the size of the object you wish to image. This can be from the literature or a pilot study. This will inform the minimum sampling rate that you will need for your sample. The Nyquist sampling rate is the ideal sampling density for a given optical configuration. The SVI calculator is a simple and useful tool for calculating this rate before you start your imaging. You can see the optical factors such as microscope type, numerical aperture, and spectral properties that are taken into consideration, and it is especially important if you're going to be doing deconvolution. When taking a Z-stack, be careful to image the full structure of interest by taking care of setting up the first and last slice. This can also apply in the X and Y axes as you frame your image. In this example, the user wanted to measure the width of the filaments, but you can see that it is undersampled in X and Y, and the Z-stack has cut off the top of the filament. When acquiring Z-stacks, you should always check the final image by looking at it in the ortho view. This will allow you to quickly adjust the first and last slice and acquire the proper Z-stack. While we always want to have sufficient resolution in our image, we also want to avoid oversampling. The major pitfalls of oversampling are time, as oversampling makes your imaging unnecessarily time-consuming. This might seem insignificant for a single image, but it can mean that a session that takes 2 hours could have been done in 30 minutes. Another factor to consider is the cost, as a shorter acquisition time will obviously mean your imaging costs less, or that you can do more imaging in the same amount of time. Another important thing to consider is the file size. Smaller file sizes mean less storage is required. You can do faster data handling, and it will take a shorter time to process your data. These might seem minor, but they can all add up and become more time consuming or create a problem down the line. If you think your files might be too large, it could be time to review your settings. Again, we need to think about the size of the object you want to image and use an appropriate sampling rate. For example, if you're imaging nuclei at 10x, you don't need to have a frame size of 2048 by 2048 just because it looks nicer. In this example, the user wanted to count the number of neurons. They were imaging with Eriscan, a super resolution technique, when they could have used a standard confocal mode. You can see in this table there is a significant difference in the settings, file size and sampling rate. But ultimately, they both show the same number of cells.
You should always select a microscope with sufficient resolution to image your object of interest. Without sufficient resolution, you will be unable to resolve enough detail, and increasing the sampling rate will not restore the lost information in a low resolution image. So as discussed before, poor image quality can affect your results, and this can lead to the incorrect analysis. So always use the appropriate resolution for your object.
Before beginning your imaging, you should first think about the question that you're trying to answer with these images and decide what's a more suitable route to take. Should you focus on quantity of images or the quality? The quantitative approach focuses more on generating a large number of images. This may be because there's a large number of samples or because of statistical requirements where you want to be able to be confident in your results. From an imaging perspective, what we want to focus on here is the minimum quality required. And you should always test the settings and the processing before beginning imaging a large number of samples. In contrast with the qualitative approach, you will probably generate fewer images. This approach is often used for proof of concept experiments where you want to prove a certain hypothesis. Often these experiments will require oversampling or super resolution to resolve these specific features. A good example here would be if you have some complex structural analysis that requires high resolution, but it's not feasible to do this in large numbers. Another time when the qualitative approach is appropriate is when you simply want an image that looks really nice. This could be for a paper or presentation, but that visualization can often be key in telling the story behind your project. It's also important to note that you can combine the quantitative and qualitative approach, so you're not tied in to one or the other. It's about finding a balance in your workload and in the research and answering the scientific question. Here is an example of combining the qualitative and quantitative approach into a single project. First, confocal laser scanning microscopy was used to acquire Z-stacks at multiple positions and then stitch them together to create a high-resolution image that confirmed individual cells were being stained. This is an example of a qualitative approach. We used confocal microscopy, 3D imaging, tiling, a higher magnification objective in order to be able to confidently say that these cells were being stained. However, the user had a significant number of these zebrafish that needed to be imaged, so a fluorescence wide field microscope was used for the rest of the samples as the imaging was faster, the modality was less harmful to the fish, and the imaging was easier to set up. So the images were lower resolution, but made it a lot easier to image a lot more of them. And the key thing is that the wide field images were still suitable for the follow-up analysis. However, the confocal images were needed at the start to confirm the staining was successful.